I think that's the biggest challenge in the field, how we can overcome resistance to venetoclax, and in part it's because it's very broad use right now in all the unfit population, but it's also moving into the younger fit populations so far in the setting of clinical trials. So clearly this is a big question, and we also know patients who fail venetoclax, it's really difficult to salvage them, so to give them anything that will get them into remission, to get them to cure. So certainly we have to figure it out and ideally to prevent the development of resistance. So I gave a talk here at SOHO yesterday where we presented our preclinical data and also analysis of the clinical trial, trying to understand the mechanism of resistance. So what we know is that there are two major mechanisms of resistance. One is genomically defined. So one is mutations in what we call signaling pathways, such as FLT3 ITD mutation, RAS mutation, c-KIT mutations, and others. And what happens is that these mutations in the clones that are not as sensitive to venetoclax because they have high expression of MCL1. MCL1 is another family member of BCL2 protein family. And it's not targeted by venetoclax. So it's redundant with BCL2, but because it's not affected by venetoclax, cells that have this MCL1 protein, they will be selected and outgrow the sensitive cells that are being killed. So what happens is that there's selection and outgrowth of these clones, and therefore patients will eventually relapse faster. So at least for the FLT3 ITD, there's a solution because the expression of MCL1 is partially regulated by the mutation itself. And fortunately, we have the inhibitors approved, FDA approved for the specific mutation. So our research has identified that the combination of these inhibitors with venetoclax will reduce MCL1 and therefore sensitize to the venetoclax-based therapy. And there's great synergy if you do the combinations. So that went into clinical trial, and the clinical trials are ongoing, but the preliminary data suggests that this combination is highly effective. So the other more challenging subset of patients that are resistant are patients who have mutations in the tumor suppressor gene called TP53. So these patients are generally very resistant to chemotherapy or to any agents that we've tried in the past. And unfortunately, they turn out to be resistant to venetoclax as well. And these are very difficult to address, but our research has turned towards immunological interventions. Specifically, we exploited the combination with anti-CD47 antibody magrolimab based on the fact that we found that venetoclax can also induce eat me signals and enhance the phagocytosis, and magrolimab would block don't eat me signals. So they are synergistic in preclinical models, and the investigator-initiated trial that was started at MD Anderson also showed high efficacy in both p53 wild type and mutated patients. So this will be taken forward into, again, randomized clinical trials, which will tell us if this will be a solution for the subset of patients. So these are the main things. There are many other ways to get resistance to venetoclax, but at least for these two subsets, we think we identified the plausible solutions. We have to see how they play out in clinic.